Hi, my name is Shreya Pandya, and I'm a junior studying nutritional sciences in the College of Family Consumer Sciences here at the University of Georgia. Over the summer, I had the amazing opportunity of being able to research with Dr. Claire de la Serre in the Gastrointestinal Neurophysiology Lab. I was also able to research under graduate student Carolina Cawthorn, and we were able to investigate the effect of gram-positive bacterial products on nodose ganglion cultures. Before I conducted research over the summer, I didn't have a background on gram-positive bacterial products, but I knew about obesity, which is why I joined. I knew obesity was a major health concern in the United States, and that a chronic consumption of energy-rich diets actually promotes excessive food intake, which results in obesity. One aspect of obesity that's really important, especially in consideration with our lab, is that obesity is associated with an altered gut microbiota composition called dysbiosis, and it's characterized by an increase in some gram-positive bacterial products. So what basically happens is vagal afferent neurons, or VAN, which are the primary sensory neurons in your gut that transmit intake-related signals from your gut to your brain, can actually become inflamed and compromised, and thus cannot provide accurate satiety signaling, which results in obesity. Lipoteichoic acid, produced by gram-positive bacterial products, can actually activate toll-like receptors found on your glia. This action results in increasing the inflammatory neuronal response that you see in VAN. So Carolina and I were working together to determine if gram-positive bacterial products can induce an inflammatory response in VAN, and we also wanted to see what specific cell types were involved in inducing this inflammatory response, if there were any specific cell types involved at all. So the first thing we did was we were able to culture nodose ganglion cell bodies from male Wistar rats. We treated these cultured ganglion in supernatant, in which Seronococcus congengensis, which is a high-fat diet-associated microbe, was grown. We were then able to add the microglia inhibitor minocycline or the glia inhibitor fluocitrate, which was included in some cell treatment groups. We then employed qPCR to assess gene expression for specific cell types, and then we were able to assess whether LTA, a known gram-positive bacterial product, was able to induce the inflammatory response that we'd seen in the supernatant. After we cultured, we were able to find our results using qPCR, which is seen on graphs A, B, C, and D. On the graphs, the treatment was shown on the x-axis, and the fold change was shown on the y. For the bars, different letters on the bars indicate significant difference between the treatments. For example, this would be bar A, and then this has a B above it, signifying that there's a great difference between the treatment between A and B. Additionally, it's important to note that the graphs A, B, C, and D depict inflammatory cytokines, whereas LCN2 is a crosstalk marker. The first thing that we were able to see was INF-gamma, IL-6, TNF-alpha, and IL-1 beta. Their expressions of genes associated with glial crosstalk were actually increased by the treatment of supernatant. So with the addition of supernatant, we were able to see on A, B, C, and D, they had an increase in the expressions of genes associated with glial crosstalk. With LTA, which is another gram-positive byproduct we were interested in, we were able to only observe on one of the four cytokines, which was TNF-alpha. So only with TNF-alpha were we able to see a significant difference with the addition of LTA. Additionally, with LCN2, in which we were interested in the expression of minocycline and fluocitrate, we were able to see that the expression of LCN2, which is a signal from astrocytes used to activate microglia, was significantly increased by supernatant. And however, it was exacerbated by the addition of minocycline and attenuated with the addition of fluocitrate. So this is what we were able to determine with our results. In conclusion, we were able to determine that yes, gram-positive bacterial products are sensed by glial cells, which can induce an inflammatory response. And this is very important because it can compromise your neuronal function, thus inhibiting your satiety response, causing obesity as it results in a greater food intake. More importantly, bacterial-driven inflammatory responses may be a contributing cause to the loss of sensitivity in your satiety signaling. And lastly, gram-positive bacterial products have the potential to induce a similar response as LPS, which has been shown in previous papers and has been shown to reduce satiety signaling. Finally, I would like to thank everyone in the gastrointestinal neurophysiology lab for helping me in my research career. I'd also like to thank Dr. Claire de la Serre for being a phenomenal mentor in my research journey. Thank you very much.